Hello everyone, this is Doug again, and in the second part of our discussion about amino acids, we are going to talk about how to classify the different amino acids, because there are so many, what are their properties, and we'll talk about more why we care in the next video. And then also, how do we start linking these amino acids together to make like dipeptides and tripeptides, and eventually a whole bunch into making polypeptides proteins. So that's our plan for this lesson. Now, just as a quick reminder, right, when we're talking about proteins and amino acids, right, protein is the macromolecule, the polymer, right? So I'll put up here, protein is the macromolecule, it's the polymer. It's the polymer, right? Polymer meaning many pieces. It has lots and lots of pieces that repeat. And in our case, the pieces that repeat are the amino acids, right? These are the subunits, or you could say the monomers, the small pieces, though, the one piece, mono meaning one and mer meaning part or piece. And we would take all these amino acids and we start condensing them together, and as we condense them together, they become one larger piece, one larger macromolecule, and that's the protein, that's the polypeptide. Protein structures get pretty crazy. I mentioned down here that here's a protein that's drawn just in its sort of to highlight its main structural features like alpha helices and beta pleated sheets. We'll talk more about that in the next video. But proteins get pretty crazy. Once you start taking one of these segments and you start adding amino acid to amino acid to amino acid, you start to make these much larger complex protein structures. So we need to talk more about what these amino acids are like so we can predict, are they in alpha helices or beta sheets? Are they in the inside of a protein, the outside of the protein? Do they make dipeptide bonds? Do they not? Those sorts of questions. So that's what we're going to answer in this video. I think I showed you this particular diagram of the 20 structural amino acids. Again, you might see reference to 20 or to 22 structural amino acids. So structural amino acids kind of depends on where you look online or in what book. Some people say 20, some people say 22. It depends on if they're talking about like hydroxyproline and homocysteine also or not. And again, by structural amino acids, we're talking about these are the ones that go into proteins. There are lots of other amino acids that are out there that do not go into proteins. So things you may have heard of like creatine, for example, or taurine is in energy drinks. So those would be amino acids that are not structural. They do other things. Now, again, for my class, I don't care that you memorize these 20 structural amino acids, but I do want you to be able to categorize them. You can see in this particular picture that they're already being categorized into like things like polar and non-polar, and that is a very important distinction. So we want to take a look at these amino acids. I want you to be able to, when you see an amino acid, you can be like, boom, that's hydrophilic, or oh yeah, that can be phosphorylated, or yes, that participates in disulfide bonds. That's going to be our goal. So let me show you some examples. Right, so I have a bunch of amino acids drawn down here, so A, B, C, D, E, F, G, H. So I've got 8 different amino acids. It looks like, just as a side note, that I drew these all in the ionized or non-ionized form, everybody? The non-ionized form, so non-ionized. Right, these are all in a non-ionized form, right? I didn't take the H off of the carboxyl and put it on the amino. In this particular case, again, not any more right or wrong than anything else. And so when I take a look at these, right, when I want to classify them, I have a bunch of different classifications up here. I have hydrophilic and hydrophobic, for example. And that's exactly what it sounds like, right? Hydrophilic are things that are attracted to water. And so we're talking about things that are polar, right? The side chains need to be mostly polar. And so when you're looking at these amino acids, I should have said that from the top. When you look at these amino acids and you're classifying them, we're not looking at the whole amino acid. We're just looking at the side chains themselves. So that means, right, when I take a look at this, am I going to make this worse? Probably not. Let's take a look. If I am going to look at A, for example, right, I only care about the side chain. I'm only going to look at the side chain. I'm only going to look at the side chain of B. I'm only going to look at the side chain of C and D, etc. I won't highlight all of these. But the idea is I want to look at the side chain because the other pieces of all the amino acids, namely the amino group and the carboxyl group and that H coming off the central carbon, those are identical for all of them, right? All these pieces that I did not highlight, the top parts of all these molecules, they're all identical. So when I'm looking for the differences between them, I'm not looking at the whole molecule. I'm not categorizing them based on the whole molecule. I'm just looking at the side chain. So that's going to be an important distinction here, right, is that I'm just looking at the side chain. And I'll write that here, right? So for all of these, we're going to only look at the side chain. And I'll draw that right here. So only consider the side chain. That's all we're going to look at for all of these questions. And so for hydrophilic, right, we're talking about things that are attracted to water. Hydrophobic, right, of course, we're talking about things that are not attracted to water, right? So not attracted to water. Not attracted to water, or in other words, these are side chains that are mostly nonpolar. So let me erase that. Not polar, we're talking about nonpolar. So let's take a look at some of these, right? So let's take a look at A, for example. So if I look at A, I'm looking at just the stuff shaded in yellow here. I'm looking at A, I want to know, is it polar or nonpolar? So I'm going to go ahead and start putting my partial charges on here. Partial negative, partial positive, partial negative, and a couple partial positives on those two H's. So I'd look at this and I'd say, oh, and I forgot one, sorry, partial negative here, partial positive here, partial positive here. Just looking at that, that's a lot of partial charges for this little area. I only have 2 nonpolar carbons, but I have so many partial charges. So I would definitely say that A, for example, would be a hydrophilic amino acid. Same thing if I look at B here, right, partial negative, partial positive, negative, and positive, that also is overall, has charges all over that, that region right there. Same thing for C. If I did C, C is a little bit of a closer call, right? Partial negative here, partial positives on those, partial positive on that carbon right there. So here I have 1 polar functional group, but 1, 2, 3 nonpolar carbons. So that's still, right, I'm still using my kind of 1 to 5 rule. For every 1 polar functional group, it can take in 5 nonpolar carbons. I'm going to kind of stick with that general rule of thumb. So I'd say overall, that C here, lysine, also would be considered polar. And now if I go and I look at D, I don't see any polar bonds at all. It's only C's and only H's. So this would all be non-polar. So I would say that for D, right, since this is all non-polar, it's not going to attract water. So this would be my first example of a hydrophobic amino acid, right? There's no partial charges in that side chain. Same thing if I look down at D here, right? D also has a side chain that only contains C's and H's, as does F only has C's and H's. So that means I know that those two are also going to be hydrophobic or nonpolar, excuse me. Sorry, I got to erase this here. It takes a second for some reason. There we go. So D, E, and F are all nonpolar. G, if I look at G, I see that I have a polar bond in there, right, because I have an OH. So I've got a partially positive hydrogen, a partially negative oxygen, and a partially positive carbon here. It's got an OH on it. There's only 1 nonpolar carbon, so I'd say that that is hydrophilic. And then lastly, so is H. So G and H, right, because H has a SH, so that's another polar bond. So I can divide all of these into hydrophilic and hydrophobic, and that's just based on is that side chain mostly polar or mostly nonpolar. Now, when I go to something like is it acidic or not, acidic or basic, all we're talking about with this is that we're looking for the acidic functional group, which is the carboxyl, right, the carboxylic acid. We're looking for a carboxylic acid in the side chain. So if you see a carboxylic acid in the side chain, then that means that residue, that amino acid residue, that side chain is an acidic side chain. A does not have a carboxylic acid. B does have a carboxylic acid, right? Here's my carboxyl. There's my carboxyl there, right? C-double-bond-O with an OH. So I'd say B would be acidic. And I don't see that in D or E. I don't see a carboxyl in any others. G has an OH, but it's not attached to a carbonyl, so it's just an OH. And again, you'll notice, right, that I am only looking at, I didn't finish my thought, at the side chain, right? I'm only looking at the side chains here. I'm not looking at the whole amino acid, because otherwise all of them would have carboxyls, and you'd say all of them are acidic, right? They all have a carboxyl in them, just not all of them have a carboxyl in the side chain. When we're talking about basic amino acids, now what we're looking for is the basic functional group, or in other words, an amine. We're looking for an amine in the side chain. Right, so I'm looking for something with nitrogens or like an NH2 or something like that. And if, sure enough, if I look at A, boom, here's an amino group. There's an amine. So I know that right off the bat that A would be a basic amino acid. I look at C. C's also got an amino group. So I know that C's also a basic amino acid. And then nothing else has a nitrogen in them. So I don't have any other amines. So the only basic side chains would be found in A and C there. Then I got kind of some kind of very specific ones here, right? So can it be phosphorylated? What I mean by can it be phosphorylated, by phosphorylated we mean can we condense, can we condense phosphate with the side chain? And by condense, we mean like condensation, like we talked about in previous videos. So what we're looking for is we're actually looking for a hydroxyl, right? We're going to attach it, a phosphate to a hydroxyl group. And that's because, right, we said that when you do condensations, you attach an OH and a partially positive hydrogen from the other thing. So we're looking for hydroxyls. That's what this is really talking about here. Looking for a hydroxyl group. That's the key thing. And again, just the side chain. So not B, right? B has an OH, but that's in a carboxyl, not a hydroxyl, so that doesn't count. If I look at C, D, E, F, G, so G does have a hydroxyl. G does have a hydroxyl, so G could be phosphorylated. That's one of the amino acids that can be phosphorylated. So if you have a protein that gets phosphorylated, gets tagged by a phosphate, that means that there must be an amino acid right there that most likely has an OH on it, has a hydroxyl on it. And then SH doesn't count for being phosphorylated, so H is out. So now what do I mean by can it form a disulfide bond? Disulfide, right, just kind of as the name implies, disulfide, we're talking about two sulfurs. And so obviously we must need some side chain that has sulfur in it, which the only one in this case is H, right, because here we have a sulfur. Here we have a sulfur. We'll talk more about this when we look at actual protein structures, but let me just mention it now. What we're looking at here is that when you have two side chains, I'm going to use R as just like whatever. When you have two side chains that have an SH group in them, the idea, right, so this one R here and this R here would be from different amino acids. What can happen is that those two side chains can actually combine, and the two S's can attach to each other and then basically make the equivalent of H2. And that is called the hydrogen, that's called the disulfide bond. So that would be your disulfide bond right here. Sorry, that looks like a 5. This is an S, right? One sulfur and another sulfur makes the disulfide bond. This would be considered the disulfide bond. This is an SS attached to one another. And we'll see that show up in the tertiary structure of proteins in the next video. But I want you to just be aware of there are certain amino acids that can do this, namely only one, which is cysteine. But that's what you're looking for the disulfide bonds. You got to find something that has sulfur in it. So that would only be H in this case. And again, I don't expect you to memorize all the names and stuff, but you should be able to look at an amino acid structure and be like, yeah, that's hydrophobic, that's not, that's an acidic one, that one could be phosphorylated, etc. So that's what I want you to know, that's how I want you to be able to classify these amino acids. Now, as I keep kind of alluding to, right, we're trying to get closer and closer to polypeptides, right, trying to get closer and closer to our proteins. And in order to get to our proteins, right, we got to start adding up. We need to start adding these amino acids to one another, right? We need to start attaching them. So when you have a dipeptide, right, that stands for 2. So you're talking about linking 2 amino acids together. When you have a tripeptide, you're talking about linking 3 amino acids together. And so what we're doing here is we need to start linking these amino acids into longer and longer chains to make proteins. And so we can start to look at our very large macromolecules, these really important proteins here. And so I have 1, 2, 3 amino acids here drawn. I have alanine, leucine, and phenylalanine. And what I'm going to do here is I'm just going to condense these 3, right? So I'm going to make a tripeptide. So I'm just going to circle off my elements of water. So I've got an OH and an H. I've got an OH and an H. So I'm going to make my elements of water. Actually, let me do this in blue just so it's a little more obvious. So, yeah, like I said, I have my OH and H and my OH and H right here. That gives me 2 water molecules, right, because I did 2 condensations, and this is condensation. I did my condensation. But now I have to redraw this molecule here, right? So I'm just going to try to keep all of these pieces here. So I had H2N. I had that 1 carbon group, and now I have another nitrogen. This amino acid has that V-shaped thing in the carbonyl, and then another nitrogen with an H, and then lastly that big 6-membered ring for the phenylalanine part. And so this would be my tripeptide. All right, so I've drawn my tripeptides. I went from 3 amino acids, right? These were the subunits. So subunits. So it's 3 amino acids. And now I have my macromolecule, which is now one tripeptide. I have one tripeptide, right? And it's tripeptide because I took 3 amino acids and combined them together, right? I still have those 3 things. And what you're going to start to see as you're reading the book and stuff, you refer to the peptides, you say them in name in their order. So this would be alanine, valine, phenylalanine, tripeptide. Phenylalanine, tripeptide. And as you can imagine, if I wanted to keep on going, I would just keep on adding another amino acid, another amino acid, another amino acid. And this is how proteins get made in the ribosomes inside your cells. You just keep on adding pieces to them. As you start to make a large protein, they do have an end to them. They have what's called the N-terminus and the C-terminus. The N-terminus is kind of exactly what you would imagine. That N-terminus is the side of the protein that ends in an N. So this would be the N-terminus, this end of the protein. So this would be the N-terminus. Right, it ends with the nitro group, the amine group. And then what we're also going to see here is the C-terminus, and that's the part that ends in the carboxyl. So as you also have the C-terminus, which is where the carboxyl is. That's the other end of the peptide chain. So this would be the C-terminus. And it's the C-terminus is where the protein keeps on growing. This is where the protein continues. So that's that. The 4th amino acid that I'd add would start on that side, and then the 5th would add to that side, and you'd keep on going in that direction. The growth of the protein is in this direction to the right. The first amino acid that got added, alanine in our case, that was the first thing at the N-terminus, and then it builds from there. It has a directionist, just like DNA and RNA have like 5' and 3'. Polypeptides have an N-terminus and the C-terminus, so you just kind of build from there. And I'll show you the N-terminus and the C-terminus more in future things. But you want to be able to identify them. It's pretty easy because, again, the N-terminus is where the N is, the nitrogen is, and the C-terminus is where the carboxyl is. So that's where they get their names. So these are just small little peptides, right? A dipeptide or tripeptide. There are pentapeptides and hexapeptides and stuff like that, but we want to move on to the big boys next. So the big polypeptides, the big proteins, where their structures get really crazy really fast because they start to fold and form these little structures called alpha helices and beta sheets. And so that's going to be in our 3rd part here of this little series. We'll look at protein structures. We'll take a look at the primary, secondary, tertiary, and quaternary structure of proteins. So if you have any questions or comments, go ahead and leave them down below, and happy studying.